Everyone, welcome to my channel, and today come with another video. And before this I explained a different topic about that related to enzymes, and you obviously check out that topics that are actually the Land-Weber bar plot or double reciprocal plot, Michaelis-Menten equation, and coenzyme, enzyme inhibition. Different topics I taught in my previous videos. You check out and follow after that this one. Okay, so let's start. Today's topic is actually we learn about the allosteric modulator or the allosteric modulation. What is the allosteric modulation? Actually, enzyme has some enzymes. They have active site. Obviously, they have present, but another site. This is called the allosteric site. This is present in that type of enzyme, and this allosteric site actually helpful for the binding of the ligands, and these ligands are called the allosteric modulator, and binding of these ligands to the enzymes allosteric site, this helps actually the binding of the substrate with the enzyme, or it helps to form of the ES complex or the enzyme substrate reactions. Clear? So here this is the enzyme modulation, and here the what is written here, the activity of the enzyme may be either enhanced or inhibited. So that when the ligand bind to the not the active site only for the substrate, so this is the allosteric site. Okay, so when the ligand binds to the allosteric site, the reactions must be depressed or stimulated. Okay, must be depressed or stimulated or inhibited. You can say that enhanced or inhibited. So this is called the allosteric modulation. Actually, these ligands are the low molecular weight. They have very low molecular weight, and they have the specific site of the enzyme other than the active site. Okay, I just told you that the active site only for the substrate, but the ligands, they cannot bind the active site. They actually bind the specific site. This site is called the allosteric site. Clear? Okay. So this is called the allosteric site, and these ligands are called the allosteric modulators, and the enzyme that contains this allosteric modulation, these are called the allosteric enzyme. Okay, and now we learn about the characteristics of the allosteric enzymes, or the what is present within this allosteric modulation. First one, the allosteric site is different from the active site. We just learn here, okay? And second one, this site may possess only one allosteric site for binding the modulator. Obviously, this site must have one site or one position or one location for binding one ligand. Okay? And third one site is there for binding minus or plus modulator. If the minus ligand bind there, then plus ligand cannot bind there. Okay? If the plus ligand or the positive ligand bind there, so the minus ligand, they have no chance for binding that site. So there is the site only for one ligand binding. Okay? The last one, fourth one, the allosteric enzymes are the oligomeric. Why the oligomeric? There is more than one substrate can bind. That means they have only one allosteric site, but more active site. Okay, more active sites for binding of the different types of substrate. Not only one substrate. Here, more than one substrate can bind with this type of enzymes. Now we learn what is the mechanism, that means how this allosteric modulator, they control or they regulate the enzymes and the substrate binding capacity or activity. First one, when the modulator bind to the allosteric of the enzyme, allosteric site of the enzyme, the three-dimensional conformation of the enzyme changes. That means in such a way the dimension or the conformational dimension that changes, that no one can bind without particular substrate. Okay, particular substrate that is only can bind with that type of enzymes in such a way the conformational of the dimensional that changes of the enzyme, that particular enzyme without particular substrate that cannot another substrate bind with this type of enzyme. It is very specific and it is very particular. That means any substrate cannot bind to this type of enzymes. And the last one, the binding of the substrate to the active site is stimulated or depressed. So, if the positive allosteric modulator or the ligand that bind with the allosteric site of the enzyme, that means it helps the stimulation of the binding of substrates. Okay, but if the negative allosteric modulator that bind with the allosteric site of the enzyme, in that situation the reaction must be depressed. That means the substrates they cannot bind, or they try to bind, they train to bind with the enzyme, but the reaction must be depressed. Okay, so this is the allosteric modulation and the characteristics. After this, what type of situation is created here, we'll learn now. Okay. Okay. So this is the allosteric modulation. You can see here the positive and the negative modulation. The active site enhanced for binding the substrate positive, that is example citrate in case of the enzyme acetyl CoA carboxylase, and this is a positive modulator. Okay, and it helps in the increasement of the catalytic enzymes in case of malonyl CoA to the acetyl CoA, the conversion of the malonyl CoA to the acetyl CoA. And this is the positive, and here this is the negative. The active site inhibited for binding the substrate, and this is one kind of example, that is the palmitoyl CoA, that is the minus or the negative modulator that suppresses the activity of the acetyl CoA carboxylase. Okay, the two types of modulators here, the positive and the negative modulators. One is the that enhance the reactions, increasement the reaction activity, and another decrease the reaction activity. Now, here we learn about the K enzyme and the M enzyme. What is the K enzyme and the M enzyme? In case of K enzyme, the positive modulator, when the positive modulator present in the medium, then the Km value decreased. Okay, and when the negative modulator present in the medium, then the Km value increased. But in the both cases, the Vmax value that remain unchanged. And this is the example, phosphofructokinase. Okay, and this is the graph you can see here. What is that? In case of K enzyme, the Km value, you see that Km value increased when the modulator is negative. Okay, when the modulator is negative, then the Km value increased. But when the modulator decreased, you see that the Km value also decreased. Okay, and the Vmax, that is same. Okay, half Vmax and the Vmax that remains same in the three situations. Next, and this Km value, this is the three steps we have taken here, or we can see here the Km dash, Km and Km double dash. And in case of M enzyme, no changes in Km values. You see here three curves or the three graphical structure you see here. Here the Km value in three situations are same. Okay, and but you see here the Vmax value changes, or in that situation, this is the half V3, this is the half V1, and this is half V2. Okay, so in presence of positive and negative modulator, Vmax rises. When the positive modulator present, then Vmax rises. When the negative modulator present, then Vmax falls. Okay, but Km remains same. So these are two types of enzymes. One is K enzyme and another is the M enzyme. I think you can understand. I'll just you summarize this K enzyme and the M enzyme again. That when the plus or the positive modulator present, then Km value decreased. but when the negative modulator present, then the Km value increase in case of K enzyme. But in case of M enzyme, when the positive present, then the Km same, and negative present modulator, then Km same. What happened in case of Vmax? When the positive or negative modulator, anyhow, anyone present, Vmax remains same. But here you see the Vmax rises when the positive modulator present in the medium, and Vmax falls when the negative modulator present. Okay, and these are the two examples. So this is the allosteric modulation. And what is the allosteric regulation in case of enzyme? It is very important for binding with the enzyme because it changes the conformational structure of the enzyme, and that helps for the binding of substrate. Not only one substrate, here more than one or two substrate can bind. And it helps either enhancement of the reaction of the enzyme substrate complex or the formation of the enzyme substrate complex, or it depressed or inhibited the binding of or the formation of the ES complex. Okay, so this is the actual allosteric modulator, and this is the role of the allosteric modulator. Okay, so I think you can understand this. Okay, and you check another videos from my channel, and before that you will check another videos, because I always try to upload different videos, yet I have very less time for uploading the videos. But if you subscribe this my channel, then you can get the notifications of any videos when I upload in the channel. You can get the notification. Okay, so please do subscribe and share my channel for getting the this type of many knowledges. Okay, thank you.